Okay, so I would like to thank Dr. Moffitt for the introduction and also Carl for this great opportunity. Great to see you guys. Have you guys ever had this question that, can I eat these delicious donuts and not gain weight, right? If we know what really happened to those sugar we eat, probably we can find the answers, right? Sugars, once we eat, they turn into glucose in our mouth and gut. And then cells uptake those glucose to process through the glucose metabolism to produce various essential molecules for us to survive. Not surprisingly, glucose metabolism is directly linked to many diseases such as cancer, dementia, heart disease, aging, not to mention diabetes and obesity. But today, most of what we know about the glucose metabolism is from the studies with enzymes in the test tubes, instead enzymes in cells. While we've learned a lot from the previous research, but how the glucose metabolism really works in the living cells has remained mysterious. So today, I am here to tell you that my research at UMBC has discovered these mysterious mechanisms with our collaborators. This is the map showing networks of the metabolic pathways. There are more than 3,400 enzymes involved in this. Every single enzyme performs very specific chemical reactions. And in many cases, series of the enzyme reactions are required to get their job done. And one of them is the glucose metabolism, as shown in the red box. If we look at it more closely, glucose metabolism has two pathways. One is the glycolysis that produces pyruvate from glucose, gluconeogenesis that forms glucose from the pyruvate. In glycolysis, there are 10 enzymes, and then they perform the series of the enzyme reactions. Then let's think about how these enzymes take place. How this glycolysis takes place in the cells. All 10 enzymes exist in the cytoplasm. And it makes sense for them to organize to perform the series of the enzyme reactions like this. However, enzymes don't know how to line up, right? So in the cytoplasm, they randomly distributed. The more problem is that the cytoplasm is really crowded. Numerous biomolecules are packed in the space. So for them to efficiently communicate each other is not possible. The series of the reaction is going to be very difficult. But somehow, cells manage to have a very efficient and well-regulated glycolysis. Right? So that was the question that scientists had puzzled so long. How this glycolysis and gluconeogenesis can happen efficiently and well-regulated way, right? So to find the answers, we monitored those enzymes in the living cells using fluorescence microscope. And these are the cells, how they look like under the fluorescence microscope. Green area shows the cytoplasm. And inside the green area, as you can see, very bright yellow spots there. Those yellow spots are the compartments that are composed of multiple enzymes for glycolysis and gluconeogenesis. These compartments are not bound with the membrane, not like the nucleus or the mitochondria. And then the concentration of the enzymes in the compartments is really high. They go up to 20 times compared to their concentration outside of the compartments. Because of the high concentration of the enzyme, the communication between enzymes are much easier. and then so that they can perform the series of the chemical reactions. So basically, these compartments are the society of enzymes for the glucose metabolism, right? But then cells are three-dimensional entities, and they keep responding to the foods that we eat and stresses that we encounter. So to study them better, we built this unconventional microscope in my lab. This microscope can visualize very small features inside the living cells in 4 dimensions. So when we put our cells under this microscope, they look like this. These green little features are glucosome. A shows a whole cell. B, C, D shows the 3-dimensional zoomed-in image of those rectangles inside the A image. When glucosomes are small, submicron scale, they are sphere, as shown in D. When they are large, they are micron scale, then they have various shapes, and they constantly change, like as shown in B and C. If we look at them in 4D, sometimes we can see that they collide each other and merge to form a larger glucosomes. And then these green arrows are pointing at the merging events. And the glucosomes rapidly respond to the changes of the cellular environments. So for example, if we apply high salt concentration in their media, then while cell volumes decrease, the glucosome formation is promoted so that their amount is increased. Then the next question that we had, how these glucosomes are going to communicate with other metabolic pathways. So we specifically studied the communication with the mitochondria and glucosome. So the final product of the glycolysis is pyruvate. That is the fuel for the mitochondria. And as you know, the mitochondria is the powerhouse of the cell. So their communication controls the cell's life. So we visualized glucosome and the mitochondria simultaneously in the living cells. Green shows glucosome, red are the mitochondria. As you can see visibly, glucosomes are nearby mitochondria. With our specialized microscope, we can quantitatively measure their 3D distances between glucosomes and mitochondria. In this graph, the highest population of glucosome touches mitochondria. If we look at them in 4D, although they are really dynamic, their contact between glucosome and mitochondria are maintained. Also, the closer the glucosomes are to mitochondria, the stronger functional link between them. And the glucosome sizes are related to their function as well. This means that not all glucosomes have the same function in a cell. Their locations and their sizes determine their function. And glucose metabolism not only networked with the mitochondria, it has networked with many other metabolic pathways. So maybe at a different location with the different sizes, these glucosomes may network with the other metabolic pathway as well, like this. So I believe understanding these networks, how these networks are regulated in 4D, holds a key to find new strategies for developing drugs of many metabolic diseases. And finally, in the future, I believe that we can eat these delicious donuts, as many as we want, without worrying to getting diabetes and obesity. Thank you.